Cell cycle and cell division, mitosis. Are you aware that from a single cell all organisms, even the largest, start their life? You may wonder how such large organisms are formed from a single cell. Characteristics of cells are growth and reproduction, indeed of all living organisms. By dividing into two, all cells reproduce. Each time they divide, each parental cell gives rise to two daughter cells. The daughter cells, which are newly formed, can themselves grow and divide, due to which a new cell population is formed by the growth and division of a single parental cell and its progeny. Such cycles of growth and division, in other words, allow a single cell to form a structure consisting of millions of cells. Cell cycle. In all living organisms, cell division is a very important process. DNA replication and cell growth also take place during the division of a cell. To ensure correct division and formation of progeny, cells containing intact genomes, all these processes, that is cell division, DNA replication and cell growth, have to take place in a coordinated way. The sequence of events by which the genome of a cell duplicates other constituents of the cell are synthesized and eventually divides into two daughter cells is termed cell cycle. Although in terms of cytoplasmic increase, cell growth is a continuous process. Only during one specific stage in the cell cycle, DNA synthesis occurs. The replicated chromosomes, by a complex series of events during cell division, are then distributed to daughter nuclei. These events are themselves under genetic control. Phases of cell cycle. In culture, a typical eukaryotic cell cycle is illustrated by human cells. Once in approximately every 24 hours, these cells divide. However, this cell cycle duration can vary from cell type to cell type and also from organism to organism. Yeast, for example, is in only about 90 minutes can progress through the cell cycle. The cell cycle is divided into two basic phases: interphase, M phase. The interphase represents the phase between two successive M phases, and the M phase represents the phase when the actual cell division or mitosis occurs. It is significant to note that cell division proper lasts for only about an hour in the 24-hour average duration of cell cycle in a human cell. More than 95% of the duration of cell cycle lasts the interphase stage. With the nuclear division, the M phase starts corresponding to the separation of daughter chromosomes, and with division of cytoplasm, it usually ends. Though called the resting phase, the interphase stage is the time during which the cell is preparing for division in an orderly manner by undergoing both cell growth and DNA replication. The interphase is divided into three further phases: G1 phase or gap 1, S phase or synthesis, G2 phase or gap 2 phase. The interval between mitosis and initiation of DNA replication corresponds to the G1 phase. The cell is metabolically active and continuously grows but does not replicate its DNA during the G1 phase, the period during which DNA synthesis or replication takes place. In the S or synthesis phase, the amount of DNA per cell doubles during this time. For example, if the initial amount of DNA is denoted as 2C, then it increases to 4C. However, in the chromosome number, there is no increase. If the cell at G1 had diploid or 2n number of chromosomes, the number of chromosomes remains the same, that is 2n, even after S phase. DNA replication begins in the nucleus and the centriole duplicates in the cytoplasm in animal cells during the S phase. While cell growth continues, proteins are synthesized in preparation for mitosis during the G2 phase. Some cells do not appear to exhibit division in the adult animals, while many other cells divide only occasionally as needed to replace cells that have been lost because of cell death or injury. These cells that do not divide further enter an inactive stage called the quiescent stage or G0 of the cell cycle and exit G1 phase. In this stage, the cells remain metabolically active, but depending on the requirement of the organism, they no longer proliferate unless called on to do so. Mitotic cell division is only seen in the diploid somatic cells in animals. Against this, in both haploid and diploid cells, the plants can show mitotic divisions. M phase of the cell cycle, this is the most dramatic period which involves a major reorganization of virtually all components of the cell. In the parent and progeny cells, since the number of chromosomes is the same, it is also called as equational division. Though mitosis has been divided into 4 stages of nuclear division for convenience, it is very essential to understand that cell division is a progressive process and very clear-cut lines cannot be drawn between various stages. Mitosis is divided into the following 4 stages: prophase, metaphase, anaphase, telophase. Prophase. Following the S and G2 phases of interphase, prophase is the first stage of mitosis. The new DNA molecule formed are not distinct but intertwined in the S and G2 phases. The phase marked by the initiation of condensation of chromosomal material is prophase. During the process of chromatin condensation, the chromosomal material becomes untangled. During S phase of interphase, the centriole which had undergone duplication now begins to move towards opposite poles of the cell. The following characteristic events can thus be marked by the completion of prophase. Compact mitotic chromosomes are formed when the chromosomal material condenses. Chromosomes are composed of 2 chromatids which are attached together at the centromere. Initiation of the assembly of mitotic spindle, microtubules and proteinaceous components of the cell cytoplasm help in the process. When viewed under microscope, cells at the end of prophase do not show endoplasmic reticulum, Golgi complexes, the nuclear envelope and nucleolus. Metaphase. The start of the second phase of mitosis is marked by the complete disintegration of the nuclear envelope, hence the chromosomes are spread through the cytoplasm of the cell. Condensation of chromosomes is completed by this stage and under the microscope they can be observed clearly. The morphology of chromosomes is most easily studied at this stage. Metaphase chromosome is made up of 2 sister chromatids which are held together by the centromere at this stage. At the surface of the centromere, small disc-shaped structures are called kinetochores. To the chromosomes that are moved into position at the center of the cell, these structures serve as the sites of attachment of spindle fibers. Hence, all the chromosomes in metaphase come to lie at the equator with 1 chromatid of each chromosome connected by its kinetochore to spindle fibers from one pole, and by its kinetochore, its sister chromatid is connected to spindle fibers from the opposite pole. At metaphase, the plane of alignment of the chromosome is referred to as the metaphase plate. In metaphase, the key features are kinetochores of chromosomes are attached to the spindle fibers. At the spindle equator, the chromosomes are moved and get aligned along metaphase plate through spindle fibers to both poles. Anaphase. Each chromosome arranged at the metaphase plate at the onset of anaphase split simultaneously, and the two daughter chromatids, now referred to as chromosomes of the future daughter nuclei, begin their migration towards the two opposite poles. From the equatorial plate, as each chromosome moves slowly towards the pole, is the centromere of each chromosome, and hence at the leading edge with the arms of chromosome trailing behind. The following key events occur at the anaphase stage. Chromatids separate as the centromeres split. To the opposite poles, the chromatids move. Telophase. In mitosis, at the beginning of the final stage, that is telophase, the chromosomes decondense and lose their individuality when they reach their respective poles. The individual chromosomes can no longer be visible at the two poles as the chromatin material tends to collect in a mass. At this stage, the following key events occur. At opposite spindle poles, chromosomes cluster and their identity is lost as discrete elements. Around the chromosome clusters, the nuclear envelope assembles. Nucleolus, Golgi complex, and ER or endoplasmic reticulum are reformed. Cytokinesis. Not only does mitosis accomplishes the segregation of duplicate chromosome into daughter nuclei but into two daughter cells. The cell itself is divided by cytokinesis, a separate process at the end of which cell division is complete. By the appearance of a furrow in the plasma membrane, this is achieved in an animal cell. Dividing the cell cytoplasm into two, the furrow gradually deepens and ultimately joins in the center. However, plant cells are enclosed by a relatively inextensible cell wall. Therefore, by a different mechanism, they undergo cytokinesis. Wall formation in plant cells starts in the center of the cell and grows outwards to meet the existing lateral walls. With the formation of a simple precursor called the cell plate, the formation of the new cell wall begins, and it represents the middle lamella between the walls of two adjacent cells. Organelles like mitochondria and plastids at the time of cytoplasmic division get distributed between the two daughter cells. In some organisms, multinucleate condition arises, leading to the formation of syncytium, as karyokinesis is not followed by cytokinesis. Significance of mitosis. Usually, the equational division or mitosis is restricted to the diploid cells only. However, haploid cells also divide by mitosis in some social insects and in some lower plants. In the life of an organism, it is very essential to understand the significance of this division. Diploid daughter cells are formed from mitosis with identical genetic complement usually. Mitosis results in the growth of multicellular organisms. Disturbing the ratio between the nucleus and the cytoplasm results the cell growth. To restore the nucleocytoplasmic ratio, it therefore becomes essential for the cell to divide. Cell repair is a very significant contribution of mitosis. The cells that are being constantly replaced are the cells of the upper layer of the epidermis, cells of the lining of the gut, and blood cells. In the meristematic tissues, the apical and the lateral cambium, the mitotic divisions result in a continuous growth of plants throughout their life. Please share this video with your friends. Leave your comments below and please subscribe to my channel for more content. click that bell notification that would let you know when a new video is uploaded. Thank you so much for watching. Once again, please subscribe to my channel for more content.